genome is really big. Really, really big. If we were to stretch out your DNA, its length would be about 2 meters. And each strand of the double helix is made of 3 billion bases. So that's 6 billion bases in total in each of your cell with a nucleus. It's massive. If we were to represent your entire genome with a straight line, not all of it, however, would contain a gene. We estimate that humans have about 25 to 30,000 genes, which I'm just representing with these grey lines here. Most of the DNA, however, is what we call a non-coding region. They don't contain genes, and therefore these bits do not code for proteins. It's estimated about 80% of your genome is non-coding region, and 20% are genes. So as a recap, what genes do is that they contain the nucleotide base sequence, and the order of those bases determines the order of amino acids. And when the amino acids are joined up on the ribosomes, they make what's called a polypeptide. Polypeptides will then fold up to make the final protein. And it's the shape that the polypeptide chain folds up in to make the protein that is significant in determining its function, whether it's an enzyme or hormone. The shape is all important. So let's look at how proteins are actually made in a process called protein synthesis. That involves 2 steps, one called transcription, one called translation. What we need to bear in mind is that the DNA is inside the nucleus that doesn't leave the nucleus. It's protected there, yet the proteins that the DNA codes for are made in the cytoplasm on ribosomes. So how does that occur if the DNA doesn't leave? So if we were to zoom in to the nucleus and have a look at what's going on. Transcription, the first part of making proteins, begins when there's an enzyme called RNA polymerase, and it actually binds to one of those non-coding regions, which I mentioned earlier. That causes the double helix to unwind. Not all of it, just a part of it. Just a part where the gene is, where the RNA polymerase is bound. The RNA polymerase will then move along this part of the double helix that has been unwound. And as it does that, it will start to complementary pair up the DNA nucleotides with nucleotides that make up RNA. Now look at the base pairing. There is one subtle difference with RNA. We do not have a T. Wherever you'd expect a T to be inserted, it's replaced with a U. U stands for a nucleotide called uracil. So as you follow the RNA polymerase down, you'll notice that T in DNA is joined with an A from RNA. You'd expect that. Then the A in the DNA is not paired with a T, it's paired with a U. The rest of the complementary base pairing is normal. C always pairs with a G. T in a DNA will always pair with an A in RNA. But the A in a DNA will pair with a U in the RNA. The other difference is that the messenger RNA, which we're about to make, the RNA is short, not long like DNA, and it doesn't form a double helix, it's single-stranded. But it's now called messenger RNA. It's going to pass the message on to the ribosomes. Before we get there, each nucleotide is split up into 3 parts called a codon or triplet code. And this is how the message is read in groups of 3. And a good representation is here, where you can see the DNA helix going into the RNA polymerase at the top and the single-stranded messenger RNA coming at the bottom. So an overview. RNA is made in the nucleus and it now leaves the nucleus and attaches to a ribosome somewhere in a cytoplasm. So the genetic message from the DNA has now been transferred into the cytoplasm of the ribosome. The next step occurs, which is translation, and here you can see our messenger RNA sitting inside a ribosome. Now, how we now make a protein is that amino acids are brought into place, and they're brought into place using something called transfer RNA, tRNA, and that has a complementary triplet code to the codon. We call it an anticodon. So AUG pairs up with UAC. Now the amino acids are sitting on top. There are 20 different amino acids. They all have different names. You don't need to learn them. This first one is called methionine, MET for short. The next codon is UUA, and that pairs up with the tRNA anticodon, AAU. This one carries another amino acid, leucine. The 2 amino acids now need to join up. And once they're joined up, the messenger RNA is moved along the ribosome. And that first tRNA will be released. We don't need it anymore, but it will leave in place the amino acid it brought along. That now means there's another codon sitting in the ribosome, GUC. Think about its complementary anticodon, and that is CAG, and that brings in a different amino acid now. This one's called valine. The next step, join them together, the amino acids. Messenger RNA moves along and the tRNA is released, leaving another codon in the ribosome, AGU, and its complementary anticodon on a tRNA is UCA, and that brings in another amino acid, a different one now. So the different anticodons have different amino acids attached. And at the end of the messenger RNA sequence, the mRNA sequence, there will be a codon which complementary pairs to an anticodon on a tRNA which does not have an amino acid attached. And that is the end of our amino acid sequence. It's now released. We have what's called a polypeptide. And this will go on to form our protein. So we've just seen DNA transcribed into messenger RNA and then translated into a polypeptide. So as an overall summary, DNA is transcribed into messenger RNA using the RNA polymerase enzyme. The mRNA can now leave the nucleus. It attaches to a ribosome, and the messenger RNA is translated into our polypeptide. Let's consider what happens when there is a mutation. First of all, we'll consider a mutation in a non-coding region of the DNA, close to where a gene needs to be transcribed. We'll consider a case where the mutation actually stops the RNA polymerase from binding. If the RNA polymerase cannot bind close to the gene, it means the DNA is not going to unwind. And if the DNA is not going to unwind, the RNA polymerase cannot carry on to pair up the nucleotides to make messenger RNA, and we're not going to get a polypeptide and therefore not have a protein. The other possibility is that there is a mutation that actually makes the RNA polymerase bind really easily, and this will cause more proteins, more polypeptides to be made, and therefore more proteins. Too many proteins can be toxic to a cell and equally as dangerous as not making the proteins in the first place. But mutations can also happen in the genes. We call them gene variants, genetic variation. In this example, transcription of the DNA into messenger RNA takes place as normal. Well, I've changed one of the original DNA bases, which was a C, to a T, highlighted red. The RNA polymerase will unwind the DNA and then insert the complementary RNA nucleotides to the DNA nucleotides, just as before. We'll let this run, so it's good practice for you to think about what bases are inserted. When we get to the genetic variant T, however, we're now going to get a new RNA base. So the mutation in the DNA is reflected in the mRNA. So we now have an A where previously we had a G. And when we get to translation, the mutation will be carried through. We get the same process that we came across before, where the tRNA will bring in the complementary anticodon to the codon and its corresponding amino acid. The two amino acids join up and the tRNA leaves as the messenger RNA moves along. But this time we've now got the different codon AUC and a different anticodon UAG brings in a different amino acid. So we will now end up with a different polypeptide and consequently a different protein in terms of its shape. It may not affect its function, it may even work better, or it may affect the function to cause a medical issue. of this can be quite severe. For example, maple syrup urine disease. The protein on the left is how it should look. The protein on the right is as a result of a mutation. Very different shape. And the symptoms of maple syrup disease can be quite severe. It causes a sweet odour in the urine, but can also cause severe brain damage.